All right. So we're going to be learning about, continue to learn about genetics, but we're going to look into the structure of DNA, RNA, and proteins. So we're going to look at how the message is coded. So DNA, the full name for it is deoxyribonucleic acid. And the whole purpose, the point of DNA is to have this message for making proteins. So your whole body is made up of proteins, and DNA is the thing that is the instructions on how to make those proteins. So what does it look like? So each building block of DNA looks like this little structure here on the right. You've got a deoxyribose sugar. You have a phosphate group and a base. These nitrogen bases, so this part towards the blue and the orange would be to the outside part of the DNA, and that would form the kind of the backbone. And then the green part would be the nitrogen base in the center, and the green part would be what you would kind of almost like the letters of the alphabet. That's the letters of DNA that you would use to write the code with. There's two different base pairs that match together, and they're A and T. So if there's an A on this side, there's going to be a T on the other side of the DNA. And if there's a G, there's going to be a C on the other side. And the same thing vice versa. If there's a T, it's going to match to A. So if you were to zoom into a DNA, you'd see a double helix that's spiraled and twisted. So it kind of looks like if you took a ladder and you twist it at each end, and you form that kind of wrap with the rungs towards the center. Two different spots could be located. So a eukaryotic cell, like our cells, so you are eukaryotic, or if you were looking at a plant cell, they have a separate nucleus that contains the DNA. If it were a bacterial cell or a prokaryotic cell, the DNA would be in the cytoplasm. So it just kind of floats freely in the cytoplasm for prokaryotic cells. And there's something called the central dogma of biology, and it's basically like how does information progress in living things? And it goes from DNA to RNA to protein. DNA stays in the nucleus. RNA gets made from the DNA. Then it's sent out of the nucleus to the ribosomes. The ribosomes are in the cytoplasm. They grab the RNA, and then they decode the RNA to make a protein. So it goes through those three steps. So what does RNA look like? RNA is just a single strand with the base pairs kind of floating towards the outside. And if you look at the base pairs, instead of A, G, T, and C, we've got U. It's uracil. So one base pair that's different. The purpose of RNA, it's rewritten instructions on how to make a protein. And the purpose and the reason that you would do that is that you don't want any damage to happen to the DNA. It's kind of like it's the most precious part of the cell. It's the one thing that you can't change. So you keep that safe, locked away in the nucleus at the center of the cell. And then what happens is you just read that signal. It's kind of like if you had an original copy of some document and you wanted to send that document out. Instead of sending the original, you run off a copy with the copy machine, and then you can send that document out. And if the copy gets broken down or damaged, it's not a big deal. You still have the original you can go back to. So RNA gets its name because it has a ribose sugar. So deoxy just means it's missing an oxygen, or it has less oxygen in a part of it. Ribose is the type of sugar. It has a phosphate group as well, connecting, so it's alternating ribose sugar, phosphate, ribose sugar, and then the nitrogen bases. And then in the RNA case, A matches to U and G matches to C. Single strand, eukaryotic cells, RNA can be found in the nucleus and in the cytoplasm because it gets sent out. And prokaryotic cells, remember, there is no nucleus, so just like the DNA, it's going to be found in the cytoplasm. And then there's a couple different RNAs with different jobs. So the messenger RNA is the copy, like the copy from the copy machine. It gets made from the DNA and then is passed out somewhere else in the cell. Then there's another form of RNA called tRNA, and that's for carrying amino acids in. So the tRNA grabs onto an amino acid, carries it over to the DNA strand, and then once the amino acids start to bond together, it releases and goes find another one. And then the very last type of RNA is ribosomal RNA, and basically all that is it's each ribosome is made up of ribosomal RNA. And if you think back to our cracking the code analogy, the tRNA, which is responsible for getting amino acids in the building blocks of protein, it's in on the message. So it understands. It's like we were talking about Navajo Indians. The tRNA has got that Navajo signal. The messenger RNA is coded. It's got the Navajo signal. And the ribosomal RNA, it also has the Navajo signal. So all three of those can read the RNA signal, and they work together and form different functions for making proteins. Okay, so that was the last slide. We went through a lot of information, and what's going to happen is that we're going to go through all of this in more detail in class and give more time to practice it because there's basically three parts: the structure of DNA, structure of RNA, and then how proteins are made. All three of those have a lot of information, so that's what we're going to focus on when we get back in class. All right, see you back in class.